Welcome back to the deep dive. Today we are taking a fascinating microscopic look at the urinary system.
Hmm.
We often think of the kidney as, simply a waste disposal unit, but that doesn't even begin to cover it.
Not even close.
This humble bean-shaped organ is a physiological masterpiece responsible for essential waste management, yes, but also a vital endocrine powerhouse that manages an astonishing 25% of our entire cardiac output every single minute.
It's a remarkable level of dedication for an organ of that size.
Yeah.
And, the mechanical efficiency is only half the story. Its sophisticated hormonal signaling and homeostatic control make it absolutely indispensable.
Absolutely. So our mission today is to give you a shortcut to being well informed about the structural foundation of this system. We've taken the chapter on the urinary system from Histology, a text and atlas, ninth edition.
Right.
And we're extracting the most important insights. We're guiding you, the learner, through the structures from the gross anatomy right down to the molecular filters and specialized ducts, reinforcing key concepts in a way that is clear and easy to visualize.
We'll cover every major structure and mechanism from the architecture of the glomerulus to the clever dynamics of the countercurrent system. We're translating the detailed descriptions found in the source material, the visual evidence of histology, into clear functional concepts so you can see why the kidney is the undisputed master of internal regulation.
Okay, let's start with the basics of what makes up the system. We have the paired kidneys, the paired ureters that carry fluid away, the urinary bladder for storage, and the urethra for eventual discharge. Structurally, it sounds simple, but functionally, it's mind-bogglingly complex.
It is, and the complexity is really rooted in the kidney's core function, maintaining homeostasis. It has to constantly, police the extracellular fluid. The kidneys are masters of conservation. They conserve necessary fluids and electrolytes while simultaneously disposing of metabolic waste.
And crucially, they are the regulators of blood pH. They maintain a constant plasma acidity by regulating the acid-base balance, deciding whether to excrete excess hydrogen ions, which makes the urine acidic, or retaining or excreting bicarbonates, which helps balance out the body's metabolic load.
Exactly. Everything feeds into managing the volume and composition of our extracellular fluid. Metabolic byproducts like urea from protein metabolism, uric acid from nucleic acids, and creatinine from muscle, they're constantly being dumped into the bloodstream, and the kidney acts as this high-speed filter to remove them.
And this demand for high-speed, high-volume processing is why the kidney is so highly vascular. That 25% cardiac output figure is huge. It ensures that the entire blood volume can be filtered multiple times per hour.
Right, and the filtration process itself is like a sequential factory line. Blood first arrives at the filtration apparatus inside the glomerulus. This initial step separates water and small solutes from blood cells and large proteins, and this results in the glomerular ultrafiltrate, what we call primary urine.
This primary urine, which is massive in volume, then enters the complex tubular system. And this is where the aggressive modification happens. It's a two-way street. Massive selective resorption, taking back almost everything valuable. Glucose, water, necessary salts.
And then specific secretion.
Exactly. Specific secretion, which is actively pumping remaining wastes and toxins directly into the tubular fluid.
So the final urine is this tiny residue, vastly reduced in volume, concentrated, containing water, necessary electrolytes, and the waste products. This is what the ureters convey to the bladder for storage and eventual discharge.
But as we noted, the kidney isn't just a plumber, it's an endocrinologist. Let's highlight the three critical hormonal products, starting with erythropoietin, or EPO.
Right, EPO. It's a glycoprotein hormone, and we know it's primarily synthesized and secreted by the endothelial cells associated with the peritubular capillaries, located specifically in the renal cortex.
So when blood oxygen levels drop, whether that's due to altitude or anemia, these cells sense to hypoxia.
And that signal travels to the bone marrow, where EPO acts directly on erythrocyte progenitor cells to ramp up red blood cell production. It's the kidney's direct way of responding to low blood oxygen by increasing the oxygen-carrying capacity.
And this is a major clinical nugget. Recombinant human EPO, rhEPO, has become a mainstay treatment for patients suffering from anemia related to chronic illnesses, most notably end-stage renal disease, where the damaged kidneys simply can't produce enough of the hormone naturally.
That's right. The second key player is renin. This is an acid protease produced and secreted by these specialized juxtaglomerular cells. Renin is the spark that lights the fire of the entire renin-angiotensin-aldosterone system, or RAAS.
So renin's job is simple. Cleave a circulating plasma protein, angiotensinogen, to create angiotensin I.
And that sets up the cascade that ultimately controls blood pressure and fluid volume, which we'll definitely detail later when we look at the JGA histology.
And the third major function is the kidney's role in vitamin D activation. We should probably clarify that vitamin D itself, whether you get it from the skin UV light acting on 7-dehydrocholesterol or from your diet, is just an inactive precursor.
It is. It requires a two-stage chemical makeover to become truly active. The first hydroxylation happens pretty quickly in the liver, yielding 25-OH vitamin D3. But this circulating compound is still inert.
The key insight here is that the kidney controls the final activation step. The second hydroxylation occurs only in the proximal tubules, catalyzed by the enzyme 1-alpha hydroxylase. This results in the highly active form, calcitriol.
And the placement of this final step in the kidney is so critical because it allows for precision regulation. When plasma calcium drops, it triggers parathyroid hormone, or PTH, which directly stimulates this alpha-hydroxylase enzyme. Phosphate levels also exert some control.
And calcitriol, the active form, is absolutely essential because it stimulates the intestinal absorption of both calcium and phosphate, which are necessary for healthy bone development and mineralization.
And here is a really powerful clinical connection. Chronic kidney disease drastically impairs this conversion. Without active calcitriol, patients suffer severe vitamin D3 deficiency, leading to impaired bone mineralization and reduced density. This often manifests as rickets in children or, reduced bone density in adults.
This is why managing chronic kidney patients often involves giving them supplements of active calcitriol or high doses of vitamin D, specifically to prevent a complication called secondary hyperparathyroidism.
Right, where the parathyroid glands continually try to compensate for the kidney's failure to activate the hormone. It just shows how interdependent these systems really are.
Okay, let's shift gears and look at the larger architecture. The kidney is retroperitoneal, tucked away in the back of the abdomen, roughly spanning from T12 to L3. It's a reddish, bean-shaped organ measuring about 10 by 6.5 by 3 centimeters.
The defining feature is that concave medial border, which is known as the hilum. This is the entry and exit point, the passageway for the renal vessels, nerves, and where the expanded funnel-like beginning of the ureter, the renal pelvis, emerges.
And if you look inside the kidney, the renal pelvis expands into the renal sinus, which is a cavity filled mostly with adipose and loose connective tissue. And of course, resting up top, encased in protective fat, is the adrenal gland.
The entire organ is wrapped in a protective capsule, but histologically, it's not a uniform structure. It has two layers: an outer layer of dense connective tissue with scattered fibroblasts, and then a distinct inner layer.
That inner layer is fascinating because it's highly cellular, packed with myofibroblasts. So these are cells that have both fibroblast characteristics, making matrix, and smooth muscle characteristics, meaning contractility.
While we don't know their precise mechanical function, the hypothesis is that the contractility of this myofibroblast layer provides some resistance against the huge volume and pressure variations the kidney experiences every day, helping maintain its structural integrity.
Now, if you cut the kidney open, you see the clear distinction between the outer cortex and the inner, lighter medulla. The cortex is where the party is. It receives 90% to 95% of the blood flow, which gives it that darker, reddish-brown color.
Yeah, the cortex is defined by its contents. It houses the crucial initial filters, the renal corpuscles, along with the highly convoluted tubules, the straight tubules, connecting tubules, and an extensive network of vessels. The renal corpuscles are the visible starting points of urine formation.
And within the cortex, you see these vertical stripes radiating out from the medulla. These are the medullary rays, also called the rays of Ferrane. There are hundreds of these projections.
Histologically, a medullary ray is an aggregation of the straight segments of the nephrons and the collecting ducts, all running parallel to each other. The regions between these rays are called the cortical labyrinths, and these areas are just jammed full of the renal corpuscles and the highly twisted convoluted tubules.
And let's clarify terminology for a second. The entire functional unit, so the nephron plus its connecting tubule and collecting duct, is collectively called the uriniferous tubule.
Roving inwards, the medulla contains the straight tubules, collecting ducts, and crucially, the vasa recta, that parallel capillary network that enables the concentration mechanism.
The medulla is organized into 8 to 18 large conical structures called pyramids. The base of each pyramid faces the cortex, and the apex narrows down to form the papilla, which protrudes into a collecting cup called the minor calyx.
And at the very tip of that papilla is a sieve-like area called the area cribrosa. This area is perforated by the openings of the collecting ducts, which here are known as the papillary ducts of Bellini, and this is where the final urine drips into the minor calyx.
So it's a very structured pathway. Minor calyces merge into major calyces, which then empty into the renal pelvis. And we should also mention the medullary zonation. The pyramid is internally divided into an outer medulla with outer and inner stripes, and an inner medulla, which simply reflects the precise positioning of different segments of the nephron.
And finally, we have the renal columns, the columns of Bertin, which are extensions of cortical tissue that spill over the sides of the pyramids. Although they contain cortical components, they delineate the divisions between the medullary structures.
Okay, let's clarify the two structural ways we divide the kidney. The lobe versus the lobule. The lobe is the anatomical unit. One medullary pyramid plus all the associated cortical tissue that sits over its base and sides. We are born with 8 to 18 of these, but they typically fuse and disappear postnatally.
The lobule, however, is the physiological unit, the renal secretory unit. It is defined by the central structure it drains, a central medullary ray, and all the surrounding cortical material, the cortical labyrinth, that drains into the collecting duct running down the center of that ray. So this makes the lobule the true functional organizational principle.
Okay, so on to the nephron, the workhorse. We have about 2 million of these per kidney, and they perform the secretory function, producing the initial urine. The collecting ducts then act as the final adjustment mechanism, determining the concentration.
It's interesting to remember that the nephrons and collecting ducts actually start from separate developmental primordia. The tubules are the secretory segment, and the ducts are, well, they're the plumbing.
So let's trace the flow. Like in figure 20.7, it starts with the renal corpuscle, the filtration ball. This is the glomerulus, that tight tuft of capillaries, encased within the double-layered epithelial cup called the Bowman capsule.
And the corpuscle has two defined points. The vascular pole is where the blood supply enters, the afferent arteriole and exits, the efferent arteriole. The urinary pole is where the ultrafiltrate leaves the capsule and enters the first portion of the tubule.
The rest of the nephron is a sequence of highly specialized pipes. The proximal thick segment, which has convoluted and straight portions, the thin segment, which is the loop itself, and the distal thick segment, again with straight and convoluted portions.
And the distal convoluted tubule ultimately connects to the cortical collecting duct, often via an intermediate connecting tubule. This entire chain is what we call the uriniferous tubule.
Tracing that convoluted course is really key. So from the urinary pole, the fluid enters the highly twisted proximal convoluted tubule, or PCT, sitting squarely in the cortical labyrinth. It then straightens out, entering the medullary ray as the proximal straight tubule, or PST, which is the thick descending limb.
This thick limb descends into the medulla, transitions dramatically into the thin descending limb, makes a sharp U-turn, and becomes the thin ascending limb. It then transitions back into the distal straight tubule, the DST, which is the thick ascending limb, and that runs back up into the cortex.
And here's a mandatory start for you, the learner. The critical structural interaction. The DST leaves the medullary ray and circles back to the vascular pole of its own parent renal corpuscle.
Right.
At that contact point with the efferent arteriole, the epithelial cells are drastically modified and crowded to form the macula densa.
Past this sensor, the tube becomes the distal convoluted tubule, or DCT, which is less tortuous than the PCT, making it less visible in histological sections. It then drains into the cortical collecting duct.
Now, a quick note on nomenclature. The loop of Henle refers collectively to the U-shaped portion, encompassing the straight proximal, the thin descending and ascending limbs, and the straight distal segments.
We must highlight the types of nephrons because their structure dictates the kidney's ability to concentrate urine. Subcapsular, or cortical, nephrons live near the capsule. They have short loops that barely penetrate the outer medulla and are less involved in concentration.
The vital players are the juxtamedullary nephrons, making up only about 1/8 of the total. Their corpuscles are deep, near the corticomedullary junction, and they feature dramatically long loops of Henle that extend deep into the inner medulla. If you need to concentrate urine, these are the ones doing the heavy lifting.
Finally, the collecting ducts. They begin in the cortex from connecting tubules, descend through the medulla, and eventually merge into the large papillary ducts, or ducts of Bellini. These ducts dump their contents into the minor calyx at the perforated tip of the papilla, the area cribrosa. It's a very organized delivery system.
Let's dive into the core of the kidney's genius. The glomerular filtration barrier. The renal corpuscle itself is tiny, about 200 micrometers, but inside there is an incredible piece of bioengineering. The filtration barrier consists of three components stacked tightly together.
The first component is the endothelium of the glomerular capillaries. Now, these capillaries are special because they are highly fenestrated, meaning they are riddled with large holes, about 70 to 90 nanometers. Crucially, unlike other capillaries in the body, these fenestrations lack diaphragms.
You can think of it like a sieve that's missing its handle. It allows large volumes of fluid through very quickly. These cells also have AQP1 water channels and secrete signaling molecules like NO and PGE2, indicating they are actively involved in regulation, not just passive lining.
The second, central, and perhaps most physically imposing barrier is the glomerular basement membrane, or GBM. This is a remarkably thick structure, roughly 300 to 370 nanometers, and it is a collaborative product of both the endothelial cells and the specialized podocytes.
Its chemical composition is critical. It's a dense network, primarily of type 5 collagen, along with laminin and highly negatively charged proteoglycans. When we look at it under specialized microscopy, it lights up brightly, confirming its dense, matrix-rich composition.
And this membrane is not just structural, it dictates health. Mutations in the type 5I collagen genes are the cause of Alport syndrome. When the GBM is defective, it becomes irregularly laminated and thickened, resulting in a filter that leaks blood and protein hematuria and proteinuria and leads to progressive renal failure.
The third layer is formed by the podocytes, which make up the visceral layer of the Bowman capsule. These are highly specialized cells. When you see an image of them, they look like little octopus arms wrapped around the capillaries.
They do. These cells send out primary processes, which branch into secondary processes, and these terminate in finger-like projections called pedicels, or foot processes. The pedicels from adjacent podocytes interdigitate like interlaced fingers, but they don't actually touch.
Right, and the narrow gaps between the interdigitating pedicels are the filtration slits, only about 40 nanometers wide. And these slits are crossed by the filtration slit diaphragm, the final, most selective piece of machinery.
The molecular architecture of that slit diaphragm is a marvel. The key structural protein is called nephron. It's a large transmembrane protein that forms a zipper-like sheet configuration, connecting one foot process to the next.
So this zipper effectively creates a self-assembling security gate. If nephron is defective due to a mutation in the nephron gene which causes congenital nephrotic syndrome, the barrier fails completely, resulting in massive, life-threatening protein loss in the urine.
We also have podocylicin covering the apical membrane of the foot processes. This sialigalic lipoprotein is highly negatively charged. Its main job is simple: repulsion. Its negative charge keeps the adjacent foot processes apart, maintaining the open patent slits.
Let's go back to the GBM and its dual function as a filter. The source material describes the GBM as having three conceptual layers, but the takeaway is twofold: physical filtering and charge filtering.
Right. The central, dense layer of the GBM is the physical filter. This collagen network restricts particles based purely on size. Anything larger than about 70 kiloday or 3.6 nanometers radius, like albumin, should be blocked here.
But the layers adjacent to the endothelium and the podocytes are rich in polyanions. These are negatively charged molecules, and these negative charges are the charge filter. They repel any circulating negatively charged molecules, reinforcing the exclusion of highly anionic proteins like albumin, even if they might be small enough to physically pass.
This dual selectivity is why the system is so robust. When we see albumin in the urine albuminuria, it often signals damage to this charge barrier, such as the reduction of anionic sites seen in early diabetic nephropathy.
And finally, a quick look at the rest of the corpuscle. The filtration apparatus is surrounded by the parietal layer of the Bowman capsule, which is simple squamous epithelium. If this epithelium starts to proliferate rapidly, forming a crescent shape, it is a key diagnostic feature of certain types of rapidly progressive glomerulonephritis.
And that crescent formation is dramatically illustrated in Goodpasture syndrome, which is an autoimmune disease. Here, the body essentially attacks its own bioengineered filter. Antibodies are directed specifically against the alpha-3 chain of type 5V collagen in the GBM.
The result is a linear deposition of these IgG antibodies along the membrane, causing rapid inflammation and failure. The syndrome is defined when these same antibodies cross-react with the basement membrane in the lungs, causing simultaneous glomerulonephritis and pulmonary hemorrhage. The microscopic presence of that crescent, a mix of fibrin, macrophages, and proliferating parietal cells, is how we confirm this rapid inflammatory attack. Now let's talk about the support staff that holds the capillary tuft together, the mesangium. This is the area where several capillaries share the GBM, usually near the vascular stalk, and it contains mesangial cells embedded in their own matrix.
Mesangial cells are the cleanup crew. Their most critical function is phagocytosis and endocytosis. They ingest and remove trapped debris, aggregated proteins, and immune complexes that get snagged in the GBM and filtration slits, ensuring the filter remains functional.
They also provide structural support, producing matrix components in areas where the GBM is incomplete. And they secrete various signaling molecules, IL-1, PGE2, when the glomerulus is injured.
And they possess contractile properties, which theoretically allows them to modulate glomerular distension in response to blood pressure fluctuations.
Wait, I have to stop you there. If these cells are contractile and they are embedded right next to the high-pressure filtration capillaries, why do the sources suggest their impact on the overall glomerular filtration rate, or GFR, is minimal? Aren't they perfectly positioned to dramatically reduce the filter area?
That's a sharp question, and it reflects the ongoing debate.
Yeah.
They are contractile, and they do contract,
Yeah.
particularly in response to angiotensin II. But the consensus is that the overall surface area they control is small compared to the total filtration surface. So while they adjust localized pressure, they are less critical for immediate bulk GFR control than the upstream afferent and efferent arterioles are. Their main clinical significance, ironically, is pathology.
How's
Well, their proliferation and increase in their number is a prominent feature in diseases like IgA nephropathy, diabetic nephropathy, and lupus. So while their normal mechanical function may be subtle, their pathological response is central to many kidney diseases.
Okay, that makes sense. Let's move to the ultimate control center, the highly strategic juxtaglomerular apparatus, JGA, located precisely where the nephron circles back to its own blood supply at the vascular pole. It has three core components.
First, the sensor, the macula densa. This is the specialized portion of the distal straight tubule. The cells here are narrow and crowded, appearing dense under the microscope.
Second, the secretors, the juxtaglomerular cells, or JG cells. These are modified smooth muscle cells found mostly in the wall of the afferent arteriole. They look different from typical smooth muscle because they are packed with secretory granules containing renin.
And third, the mediators. The extraglomerular mesangial cells, also called lysoscells, situated outside the corpuscle, connecting the macula densa and the JG cells.
The JGA's entire purpose is to manage blood pressure and sodium homeostasis through the activation of the renin-angiotensin-aldosterone system, or RAAS. This system kicks in when the body senses low sodium intake, massive fluid loss, or reduced renal perfusion.
The mechanism starts with the JG cells releasing renin into the blood. Renin acts as the catalyst, cutting the circulating plasma protein angiotensinogen to form angiotensin I.
That intermediate then travels to the lungs, where the magic happens via angiotensin-converting enzyme, or ACE, located on lung capillary endothelial cells. ACE instantly converts angiotensin I into the highly potent active molecule, angiotensin II.
Angiotensin II is terrifyingly effective. It is one of the most powerful vasoconstrictors known, dramatically increasing systemic vascular resistance. Furthermore, it stimulates the adrenal cortex to release aldosterone.
Aldosterone then acts on the kidney tubules to increase sodium and water reabsorption while dumping potassium, the ultimate result of the entire cascade: increased blood volume and increased pressure.
And the trigger for this entire system is the macula densa. It monitors the tubular fluid's sodium concentration. If the Na+ concentration drops, indicating low volume or flow, it senses this via specific apical cotransporters.
When sodium is low, the macula densa releases paracrine signals like ATP, NO, and PGE2 that signal the adjacent JG cells to dump their renin granules into the bloodstream, initiating the whole pressure-boosting sequence.
The clinical takeaway here is one of the biggest advances in cardiovascular medicine. Since chronic essential hypertension is often linked to an overactive RAAS, the development of ACE inhibitors like captopril provided a revolutionary treatment by simply blocking the conversion of angiotensin I to the active angiotensin II, effectively turning down the system's volume knob.
We've created the primary urine, now we have to modify it. The challenge is huge. We start with about 180 liters of ultrafiltrate a day, and we need to reduce that volume drastically, reclaim everything vital, and ensure the final product is concentrated and hyperosmotic.
This complex modification involves massive active and passive absorption of water, sodium, and glucose, and specific secretion of organic wastes. The foundation for success relies on establishing the countercurrent mechanism, utilizing the loop of Henle, collecting ducts, and parallel vasa recta.
The first post-filtration stop is the proximal convoluted tubule, the PCT. This is sort of the overzealous intern of the system. It reclaims about 65% of the ultrafiltrate immediately over 120 liters a day, producing a fluid that is still isosmotic with the plasma.
The histology reflects this extreme efficiency. The PCT cells are packed with specializations. You can see it in figures 20.17 through 20.19. Apically, they feature a huge, dense brush border of long, closely packed microvilli, maximizing surface area.
They have complex lateral folds that interdigitate extensively, and basally, they show what the source calls basal striations. Now, these aren't scratches. They are vertically oriented, elongated mitochondria packed into the basal processes, providing the incredible energy needed for transport.
The reabsorption engine is fueled by active transport. Na plus K plus ATPase pumps are located everywhere on the lateral and basal folds. They actively pump Na plus out of the cell into the lateral intercellular space, creating an osmotic gradient.
And this gradient passively pulls water out of the lumen. And the water moves fast thanks to constitutive aquaporin 1 or AQP1 channels in the membrane. The fluid then builds up hydrostatic pressure in the intercellular space and drives itself into the peritubular capillaries.
Solute reclamation is nearly complete here too. The PCT recovers essentially 100% of glucose and 98% of amino acids, utilizing specific transporters. Any large proteins that snuck past the filter are degraded by brush border peptidases or captured by deep invaginations and recycled via lysosomes.
The journey continues into the proximal straight tubule, the PST, which is the thick descending limb. The cells here are a bit shorter, less flashy, and their main job is mopping up the last bits of glucose that escaped the PCT, utilizing high-affinity SGLT1 cotransporters.
Next is the thin segment of the loop of Henle. We won't list all 4 histological cell types, but the functional distinction is everything. As the fluid descends into the hyperosmotic medulla, the descending limb is highly permeable to water, allowing it to exit by osmosis. This concentrates the solutes in the lumen. Crucially, there is no active ion transport here.
Then, the U-turn happens, and the ascending limb flips its function completely. It becomes impermeable to water, but highly active in pumping ions, specifically using Na plus K plus 2Cl cotransporters to drive solutes out of the lumen.
This ion pumping creates the essential condition: a hyperosmotic interstitium surrounding the loop, while the fluid remaining in the tubule becomes hyposmotic or diluted. This is the diluting segment.
A unique product of the thick ascending limb is uromodulin, also known as Tam-Horsfall protein. Beyond aiding ADCL reabsorption, it defends against UTIs and prevents kidney stone formation. Clinically, when this protein precipitates in the distal tubules and collecting ducts, it traps whatever is flowing past red blood cells, white blood cells, and forms organized structures called urinary casts.
And the detection of proteinuria, or the discovery of these casts in a urinalysis, is a pivotal diagnostic indicator of renal disease. These casts essentially give us a microscopic snapshot of the underlying inflammation.
So the fluid then enters the distal straight tubule, or DST, the thick ascending limb again, which continues separating solutes from water, actively transporting ions into the interstitium via those electroneutral synporters.
The active Na plus K plus ATPase pump here maintains the gradient. An interesting consequence is the positive charge created in the lumen by potassium leaking back out, which passively drives the reabsorption of divalent cations like calcium and magnesium.
Now we arrive at the distal convoluted tubule, the DCT. This segment, located in the cortical labyrinth, is shorter than the PCT. The cells are taller and lack a brush border, and the tubule remains relatively impermeable to water.
The DCT is functionally intense. It has the highest concentration of Na plus K plus ATPase activity in the entire nephron. It's the primary site for PTH-regulated calcium reabsorption, meaning the parathyroid hormone dictates how much calcium is reclaimed here. It also handles sodium reabsorption, potassium secretion, and fine-tuning acid-base balance.
The connecting tubule is the brief transition zone where the DCT merges into the cortical collecting duct. This small segment is crucial for potassium secretion, regulated by mineralocorticoids.
And finally, the cortical and medullary collecting ducts. These ducts are the ultimate deciders, determining the final osmolality of the urine by controlling water reabsorption. Histologically, they're easy to spot because, unlike the proximal and distal tubules, their cell boundaries are very distinct and clear.
The epithelium contains 2 major cell types. The most numerous are the pale-staining light cells, also called principal cells. They possess basal infoldings, single cilium, and shirt microvilli.
These principal cells are the primary targets of aldosterone. They have abundant mineralocorticoid receptors. Functionally, they reabsorb sodium using E channels and secrete potassium using Raman K channels. Crucially, their apical membrane contains the ADH-regulated AQP2 water channels.
The second type are the dark cells, or intercalated cells, IC cells. They're characterized by dense cytoplasm and many mitochondria. These are the specialized acid-based transporters.
Right. The alpha IC cells are the acid secretors. They dump H plus protons into the lumen using apical ATPase pumps and reclaim bicarbonate into the blood. The beta IC cells do the opposite, secreting base HgO3 into the lumen.
Since most diets result in a net acid load, we typically have more alpha IC cells, but the system is adaptive. In the face of severe acidosis, the beta IC cells can chemically convert into alpha IC cells to maximize acid excretion, demonstrating incredible functional plasticity.
We should probably emphasize a modern correction here regarding aldosterone action. While historically it was linked to the DCT, molecular studies confirm that aldosterone targets the principal, or light cells, of the connecting tubules and collecting ducts.
Aldosterone binds its receptor, moves to the nucleus, and starts acting as a transcription factor, increasing the synthesis of ENAS, ROMK, and NAB plus K plus 8T base. This takes several hours, but the net result is sustained sodium retention and potassium loss, which boosts blood volume and pressure. Let's talk about the interstitial cells. These are the supporting connective tissue matrix around the tubules and vessels. Their volume increases significantly as you move from the cortex, where it's about 7%, to the inner medulla, where it's over 20%.
In the cortex, they are typical fibroblasts. But in the medulla, they resemble myofibroblasts, and they contain lipid droplets that fluctuate with the kidney's diuretic state.
There is a critical clinical lesson here. Most fibroblasts originate via a process called epithelial mesenchymal transition, or EMT, from damaged tubular epithelial cells.
That's right.
Chronic injury causes these cells to overproduce extracellular matrix, leading to fibrosis and irreversible renal failure, a devastating condition called tubulointerstitial nephritis.
Let's focus back on the molecular mechanism that allows for water concentration. Aquaporin channels. These are small, hydrophobic transmembrane proteins that act as fast, efficient water conduits.
We mentioned AQP1 in the proximal tubules, which is responsible for bulk, non-regulated water movement. But the real star is AQP2, found in the principal cells of the collecting ducts. This channel is entirely ADH regulated. It's the gatekeeper.
And a mutation in the AQP2 gene is linked to congenital nephrogenic diabetes insipidus, where the collecting duct cannot respond to ADH, which just demonstrates its essential role.
All of this sets the stage for the countercurrent multiplier system. The kidney is a clever engineering trick for making concentrated urine. Countercurrent simply means fluid flow in adjacent structures moving in opposite directions.
Three structures work together. The loop of Henle is the multiplier. It uses active ion transport to establish the osmotic gradient in the medulla. The vasa recta is the exchanger. It runs parallel to the loops to maintain the gradient. And the collecting duct is the equilibrating device that allows the fluid to equilibrate with the hyperosmotic environment, provided the gates are open.
The mechanism relies on the standing gradient. The thin descending limb passively loses water, concentrating the fluid. The thin ascending limb, which is impermeable to water, actively pumps salt out.
And since this process is continuous, that's the multiplier effect, the interstitium becomes dramatically hyperosmotic, with the salt concentration increasing steeply as you move toward the inner papilla.
The final step is ADH regulation. ADH, or vasopressin, is the switch. When plasma osmolality increases, say, due to dehydration, ADH is released. It binds to the principal cell receptors, causing a dual response.
The short-term effect is very rapid. ADH triggers a translocation of AQP2 channels, which are stored in vesicles, to rapidly fuse with the apical membrane. This instantly makes the collecting duct permeable to water.
And the long-term effect is the synthesis and insertion of new AQP2. If ADH is absent, the gates remain closed, and we produce large volumes of dilute urine. If the kidney can't respond to ADH, regardless of how much is present, that's when we have nephrogenic diabetes insipidus.
Exactly. Finally, the role of the vasa recta countercurrent exchange. These specialized capillaries loop down into the medulla. As they descend, the blood passively loses water and gains salt, equilibrating with the surrounding hyperosmotic interstitium.
And as they ascend, the blood reverses the process. It loses salt and gains water.
This passive exchange ensures that the circulation supplies the necessary oxygen and nutrients to the medulla without destroying the critical osmotic gradient that the loop of Henle worked so hard to establish. It's a perfect balancing act.
Okay, let's trace the blood supply pathway. It's highly structured and predictable. Starting at the renal artery, it branches into the interlobar arteries, which travel between the medullary pyramids.
At the junction of the cortex and medulla, these arteries arch over the base of the pyramids, becoming the arcuate arteries. From the arcuate arteries, the interlobular arteries ascend straight through the cortex.
From the interlobular arteries, the afferent arterioles branch off, each supplying a single glomerulus. The capillaries inside the glomerulus then reunite to form the efferent arteriole. And you should notice this is unique, artery to capillary to artery.
Right. The efferent arteriole then supplies the second capillary network. Efferents from cortical glomeruli form the extensive peritubular capillary network surrounding the cortical tubules.
Efferents from the deeper juxtamedullary glomeruli, however, form the long, straight descending vessels known as the vasa recta, which plunge deep into the medulla to supply the countercurrent exchange system.
And venous flow generally mirrors this path. Capillaries drain into interlobular veins, then arcuate veins, then interlobar veins, and finally the renal vein. The network near the capsule drains into stellate veins before entering the interlobular flow.
Regarding lymphatic vessels, there are two networks. One in the outer cortex and capsule, and a deeper network draining the substance of the kidney toward the renal sinus. They anastomose extensively.
The nerve supply is predominantly sympathetic, unmyelinated fibers releasing norepinephrine, forming a renal plexus concentrated mainly along the afferent arterioles.
Sympathetic stimulation causes vasoconstriction. So, constriction of the afferent arteriole reduces filtration and urine production. Constriction of the efferent arteriole increases filtration pressure.
This relationship is highly relevant clinically. Chronic hyperstimulation of the renal nerves is a factor in resistant hypertension, leading researchers to explore treatments that ablate these nerves. However, it's worth noting the ultimate functional independence of the system. Kidneys transplanted into recipients with severed extrinsic nerves function completely normally.
Once the urine has passed the area cribrosa, it enters the collecting and excretory passages, the calyces, pelvis, ureters, bladder, and urethra. All these passages, except the final urethra, share a general organization. Mucosa, which is lined by specialized epithelium, a muscularis layer, and an external adventitia or serosa.
The defining histological feature is the transitional epithelium, or urothelium. This stratified epithelium lines the entire tract up to the initial urethra. Its unique property is that it is highly impermeable to salts and water, and it can accommodate massive changes in volume.
The cells are arranged in 3 layers. The superficial layer is the most remarkable, consisting of large, often multinucleated dome-shaped or umbrella cells. When the bladder is empty, they look bulky. When full, they stretch flat, looking squamous. Their edges form tight, zipper-like interdigitations.
And the overall thickness changes dramatically, from just 2 cell layers in the inner calyces to 6 or more in an empty bladder, flattening down to 3 layers when the bladder is fully distended.
This ability to stretch and maintain impermeability is thanks to the incredible surface specialization you see in figures 20.29 and 20.30. The apical membrane of the umbrella cells is covered by rigid, scallop-shaped structures called urothelial plaques or asymmetric unit membrane, AUM.
These plaques are formed by a crystalline array of proteins called uroplankins. This crystalline structure is what provides the impermeability, forming the absolute barrier against small molecules like urea and water.
So the umbrella cells contain a reservoir of these AUMs in structures called fusiform vesicles. When the bladder stretches, these vesicles rapidly fuse with the apical membrane via exocytosis at the plaque's hinge regions, dramatically expanding the surface area without compromising the permeability barrier.
This barrier is critical, but it's also the target of infection. Uropathogenic E. coli exploit the uroplankins, using specific adhesins to bind to them and gain a foothold, leading to common urinary tract infections.
The urothelium rests on a dense collagenous lamina propria, and importantly, there is no muscularis mucosae or submucosal layer. The muscle bundles sit directly beneath the propria.
And the muscle arrangement is distinctive too, particularly in the ureters and urethra. An inner longitudinal layer and an outer circular layer. This is the reverse of the typical arrangement found in the gastrointestinal tract, and these bundles create the peristaltic contractions that move urine along.
The ureters are long tubes, about 24 to 34 centimeters, that enter the bladder obliquely, creating a functional valve that compresses the opening to prevent reflux. Only at the distal end do they gain a third, outer longitudinal smooth muscle layer.
The urinary bladder is the large reservoir. The trigone, the triangular area between the ureteric and urethral orifices, is distinctively smooth and constant in thickness, reflecting its separate embryological origin. The rest of the wall forms the thick, interwoven detrusor muscle.
Right, and the detrusor contraction forces urine out. At the urethral opening, fibers from the detrusor form the involuntary internal urethral sphincter.
Micturition control is complex. The sympathetic nervous system dominates storage. It relaxes the detrusor muscle and contracts the internal sphincter. The parasympathetic system drives voiding. It contracts the detrusor and causes the internal sphincter to relax via nitric oxide release.
The final, voluntary control comes from the external sphincter, which is composed of striated muscle. This maintains tonic contraction via the somatic pudendal nerve, ensuring continence until consciously inhibited.
Finally, the urethra. In males, it's long, about 20 centimeters, and functionally dual, serving both systems. It passes through prostatic, which is transitional epithelium, membranous, with stratified columnar and surrounded by the external sphincter, and penile or spongy segments, which is pseudostratified columnar.
And the female urethra is short, 3 to 5 centimeters. It transitions rapidly from transitional to stratified squamous epithelium. It features small glands and a highly vascular lamina propria that acts much like the male corpus spongiosum. So we've taken a molecular journey through the kidney and the entire urinary tract. The essential takeaways confirm that this system is a master of homeostasis and filtration achieved through microscopic specialization.
The nephron is the masterpiece. 2 million filters utilizing cellular specializations, from brush borders to basal mitochondria, to reclaim essential resources and achieve massive volume reduction without energy waste.
We saw how the JGA and the RAAS established the kidney as a sophisticated endocrine regulator, linking sodium concentration directly to systemic blood pressure control via renin and aldosterone.
And we described the countercurrent system, a remarkable triumph of fluid dynamics, where active ion pumping creates an intense osmotic gradient that passively draws water out, enabling the concentration of urine.
And finally, the barrier system is capped off by the urothelium, a dynamic, impermeable tissue that uses uroplaque and plaques and fusiform vesicles to fold and stretch drastically, safeguarding the body from the final, highly concentrated urine.
As you consider this architecture, contemplate the molecular precision required by the entire process. The filtration barrier is a 3-layered filter operating at the nanoscale, balancing size restriction and negative charge to perfection. The incredible insight is that a failure involving something as small as a reduction in negative charge, something you can't even see, can lead to the catastrophic systemic imbalance known as diabetic nephropathy. It just underscores that biological function truly depends on molecular detail.
A compelling realization. Thank you for diving deep into the histology of the urinary system with us.
Until next time.